So let's begin. So as you remember, Bioinformatics for Precision Medicine is a program dedicated to training those that are interested to leverage big data and molecular precision to address pressing challenges in the future of medicine. And in the context of this program, we're going to be discussing a number of different technologies that allow researchers to understand the basic mechanism of action that underlies different diseases and treatments to those diseases. So we'll talk about analysis of next generation sequencing data and the application of that type of data and those types of technologies to the study of oncology and infectious diseases. In the context of this program, we have prepared a number of resources that include online courses, project examples, and all of that material is available on the Bioinformatics for Precision Medicine page. So let's take a look at that page and see some of the updates. So as I mentioned, Bioinformatics for Precision Medicine page has all of the resources that you would need to go through this program. You can see today's session right here on June 23rd. So we're going to be talking about processing next generation sequencing data, how we use next generation sequencing data to analyze gene expression. You can see updates, and we post here important updates on the forum. You can see that BeepShot updated some of the top performers. And then if you scroll down, you can also see that we're trying to interact with the majority of you. We really ask that you try to complete your profile as soon as possible, because the exact name of your profile is going to appear on the certificate of any course that you complete. So it's important to at least have a full name. But more than that, you can see some of the featured profiles. They've really spent some time to upload an avatar. Some of them even have links to their social media profiles. And previous participants of our programs have completed and showcased their profile to all of our participants and people who are in our network that include research labs and many universities around the world, as well as recruiters, people that are looking for bioinformaticians for hire, for teaching, and for different support functions. So if you're interested in any of those directions to continue your education and continue your developing your career, please make sure that you update your profile. All the individual courses will have a certificate, and those certificates, like I mentioned, will have your full name on them. So make sure that you have your name the way you would like it to show up on your actual profile. On your certificate, sorry. The other area that's important to notice is that we have a forum. The forum is designed for us to post updates, but also for you to ask any questions and to get feedback on those questions. Sometimes we will get that feedback during the time in between sessions, and sometimes during the sessions themselves. So you can see here that we post session recordings, top performers, as I mentioned already. And also we comment on your questions right here. So you can go to choosing your project. There was a good question. You can see here the question from Yuja Zhang that had a few questions. Is it free for a registered student to choose a project? How many can we commit at most? Is it independent or more tutorial style, similar to finishing a course? Do we have to finish that in a particular time window, or is it totally self-paced? So I just briefly wanted to address this, and hopefully after today's session, we'll have here a form to help guide your project submission. So really the project idea is to help you apply the skills that you've gained in this program to a data set of your own, or to a data set that we have not fully explored in the context of the session. So you are able to work on a completely independent project, or you're welcome to join one of the prepared projects, and one of those prepared projects can have an open ending. In other words, you start with something that is very guided, and eventually you can kind of deviate from that guided path and develop a project of your own, potentially focusing on some aspect of the project that has not been explained fully, or focusing on something that you can combine with the data, apply different methods, etc. The question about duration, so whether you have to do this in a certain time frame. So the program is designed to have approximately half of the program dedicated to interactive sessions and half of the program for you to work on your project. So we hope that you can complete at least some kind of a project in the time frame of this program. If you would like to continue to use the platform and rely on our support, as well as to get mentorship and feedback, we have a variety of options for you to continue after the program is over. You have access to our mentor network through the internship or the research fellowship. And you can also use the platform for a variety of different research tasks. And we have the option for research licensing. Okay, so just to make sure, is everybody able to see the screen? I see that Manal here is saying that he has a black screen. Bipsha, Mohit, do you guys see my screen?
Yes, Celia, I can see your screen. So,
Yeah.
For him, I can say, Manal, can you leave the Zoom meeting and join back again? Sometimes there is a problem with the app or the Zoom client you might be using. You can try joining again.
Okay, thank you. So yeah, and we'll have the recording available afterwards, so you'll be able to see if you missed any part of it later on YouTube. So again, I hope that clarifies the questions about the project, and we'll post here a form that can help guide your project direction. The other thing that I wanted to mention is that our programs are really not limited to one particular data type or type of question that you can ask. We have a variety of different programs, and we will hold an upcoming free webinar on July 1st at 10 a.m. CST on Wednesday. For those of you interested to develop personal data science skills, after you've learned the logic of analysis, you've understood how the different data sets are used to address research questions, after you've tried using some of the tools that we've made available through an interactive and user-friendly interface, you might want to continue and expand your data science skills, learning a little bit more about coding, learning a little bit more about a number of other visualization software tools like Chimera and PyMol. So we are going to be talking about a couple of programs that we have coming up. They're designed for beginners and those interested to kind of see the next step. So here's a webinar, and thank you, Bipsha, for posting that link. If you're interested to learning more about those programs and what we will be talking about, please register on the link that Bipsha posted there. After the free webinar, as I mentioned, we will have a number of upcoming programs still through the summer that you can join. Okay, so let's do a quick review of what we did last time. First of all, let me ask you, how many of you were able to try completing the courses, Introduction to Genomics and Genomics 1? So if you completed one of them, just type in 1. If you completed both of them, type in 2. Okay, excellent. So a lot of you have started, and many of you have even completed both of them. So that's really great. Okay, so let's do a quick review together and talk about DNA variation, how this data is processed, analyzed, and interpreted. So we talked about how DNA contains important information for the production of proteins, but that information is coded in genes. So we have these sequences of nucleotides, and those could have a number of different variants from the original sequence. So when we talk about the original sequence, we have a reference human genome. That reference human genome is used regularly to represent an average normal human genome. Some of those variations are not harmful, and they represent the variation that we normally have: different eye color, different height, different environmental adaptations that different people have experienced. And many of those are passed on from generation to generation, we call those germline mutations. So germline mutations are the acquired mutations, variants in the DNA that are normal to you as an individual or any species. Now, several different types, substitutions, insertions, and deletions are typically thought of as point mutations. And then we also talked about a number of larger variations, for example, copy number variation, so copies of genes, or variation of multiple nucleotides together. We also discussed what's called somatic mutations. Somatic mutations are the ones that are acquired during your lifetime after the cells continue to divide. And you can see how those accumulate over time, sometimes going over the threshold of what is the cells are able to suppress and fix. And those typically, if they occur in tumor suppressor genes or oncogenes or DNA repair genes, we have a problem. And so that problem oftentimes causes cancer or unproliferated, uncontrolled growth and metastasis. So talking about somatic mutations, we also talked about how to detect those somatic mutations in next generation sequencing data. And we discussed a number of different types of sequencing where we can look at whole exome sequencing, whole genome sequencing, or targeted sequencing. Targeted sequencing providing us with the biggest coverage, but only in specific genes that we are focused on. So when we talk about those different types of sequencing, targeted panel sequencing can contain anywhere between 40 and 400 genes. Whole exome sequencing, about 20,000 protein coding genes. And then whole genome sequencing includes the non-coding regions of the DNA and typically is used for study of not somatic, but germline mutations. So the different types of coverage are important here because we talked about how mutations in a specific cell, when we take bulk data, so a group of cells together, a whole tissue, we are not able to distinguish between sequencing errors and actual mutations. And so we are really just looking at the most abundant mutations that are present in many cells. One of the problems in cancer, multiclonality, is the fact that those cells, even if there's one cell that is going to develop into a metastatic cell or a cell that has this unproliferated, uncontrolled growth and starts replicating, the timing of data collection, the timing of sample collection is critical here because we can miss an important point at which we could have stopped the disease. But when we talked about this, we also talked about how data could be visualized, and we saw that FASTQ files have individual reads, and by aligning those reads to a reference genome, we can actually start looking at the different mutations that we discussed here and understand how those mutations appear in real data. So let me ask you a question. One of the examples that you probably saw was an example using cell line data. These are breast cancer cells. And we briefly looked at that example last time that we spoke. So as you look on this picture here on the right, what type of sequencing do you think this is? Is this whole genome, whole exome, or targeted sequencing? Okay, so anyone can answer. Take a look at okay, so a couple of you are thinking targeted. Okay, and you can also explain why. Why do you think this is targeted? So many of you actually think that this is targeted sequencing. Uh, so yeah, why don't you explain why you think this is targeted sequencing? What makes you think that this is targeted sequencing? Short read aligning to specific region, targeted or exome. Okay. So it is whole exome sequencing. Why do we know that? We know that indeed the reads are not aligned everywhere in a uniform way. We only see them in specific regions. Now, how do we know that this is actually exome sequencing? Down here, you can see the individual exons. Exons are the portion of gene that produce a sequence that will later be a part of the mRNA. So these are protein coding genes. Protein coding genes have exons and introns. Introns are not expressed, so they're not transcribed into mRNA, they're spliced out typically. And so when we do whole exome sequencing, we do not sequence those regions. So as you look down here, right, these are exons, and you can see that the coverage of reads is only within exons. Okay, which gene is this? This is the P53 gene. You can see right here, TP53, and it has many different transcripts. So we'll talk about that today. The TP53 gene, one of the important aspects of this gene is that many cancers are associated with a specific mutation or a number of different mutations within this gene. And so to look at the specific mutations within the TP53 gene in those cancer cell lines, we looked at a number of different positions using integrated genome browser. And typically what you would want to do is produce a somatic mutation analysis, so analysis called variant calling. And the variant calling produces a file called VCF, a file that you can actually visualize in integrated genome viewer. So this is an example. And those are the two different pipelines that we did. One pipeline for visualizing how the reads align to the reference genome, and another one for somatic mutations. So have both of you, or sorry, have all of you tried both of these pipelines to see the inputs and the outputs of these two pipelines? Okay, so if you have not, I recommend going through the demo, just like we did last time, but also you can do this on your own time. Now, has anyone had any issues running these pipelines? Okay, a review of these pipelines is in the Genomics 1 course, so you're welcome to take a look at that course. If you don't want to go through the course itself, we have some demo pipelines that you can just run through, right? So this is an important part of really understanding how genomic data, next generation sequencing data applied to the study of genome is used. And here is just an example of looking at what's in the data, right? That's the first visualization, right? Let's look what's in the data. You really understand the complexity of the data. Many chromosomes, many different genes, many different positions that you potentially might be wanting to look in. And here, an ability to detect only somatic mutations. So those somatic mutations allow us to detect specific variants that are present in only the cancer or the tumor sample by subtracting everything else that is normal to that person that represents the germline mutations. Okay, and so let's think a little bit about how this data might be used in an actual clinical study. So here is a discussion of challenges to diagnostic use by clinical genome and exome sequencing, right? So a lot of people today are thinking about exome sequencing, next generation sequencing in the clinic. How is it actually used, and what kinds of challenges arise as a result of these different steps, pre-analytical, analytical, post-analytical. So the initial testing strategy is one of the major areas. So you can, for example, select specific samples, and you have to target specific genes, or think of how much coverage you would like to have in your samples. So here we have a number of different examples, right? So in the case of breast cancer, for example, we all know that the BRCA genes are contributing to the risk of development of breast cancer. But how many of the BRCA genes have a particular mutation, and how many cells in the sample have that mutation, will dictate how much coverage and how many specific genes, BRCA1 or BRCA1 and 2, or additional genes in the BRCA pathway, or other, collections of genes, right? So those have to be determined before you actually generate the data. After the generation of sequencing data, there are a lot of issues with false negative results. For example, regions that cannot be sequenced will not generate variants for downstream analysis. Many times when we do this kind of sequencing, there are specific regions that will have more coverage than others just because of the nature of the sequence. So developing a strategy for good coverage for all of the regions that are significant for downstream analysis is a big challenge before you actually start analyzing, right? Providing enough coverage and having all the regions covered in an equal manner. Then there's a step of short read alignment, right? We saw how those reads could be aligned in different positions. And we also saw how some of the reads are actually not aligned at all. We will have unmapped reads. So having reads aligned to the correct region, having enough reads in that correct region is critical for us to actually calculate how bases change from the reference genome. Genotyping. So certainty decreases in regions with low coverage, which causes low confidence genotype calls that are discarded during analysis. There is a process of filtering, right? As we have this lower coverage in specific regions, some of those will be considered insignificant. But are they really insignificant, or are they called insignificant because we have limited amount of data, right? That's another challenge. Annotation. It's difficult to annotate many of the mutations. We just don't know enough about annotation. Sometimes we have a variant that has not been studied. It's a rare variant, and we don't know what it means. And typically, when we don't know, we just ignore it. We discard it, right? And so, many different analytical dry lab, so to say, challenges, as well as post-analytical. Sometimes frequency of mutation, sometimes mutation in a specific position, sometimes it's combinations of mutations together that actually play a role in the development of risky conditions or the progression of disease. So there are many different challenges in application of this type of next generation sequencing technology to actual clinical use. I recommend continuing to learn about these different types of data, and also think about other types of data that could help us interpret what is really going on beyond the level of the genome. So we have a number of these different courses. If you have not been able to start the Introduction to Genomics or Genomics 1, I think this is really important in the context of this program to be able to complete at least those two. Okay, so do we have any questions about analysis of genomic data, somatic mutations, germline mutations, and next generation sequencing data as it applies to genome and the study of genes in the DNA? Okay, if you have any questions, please either type them in the chat, or you're also welcome to unmute yourself and ask. Okay, if you have any questions, please make sure that you post them on the forum. If you would like those to be addressed in detail in the next time that we meet, I'll be happy to see those there and address those in detail. Okay, so today we want to talk about processing next generation sequencing data to study RNA-seq, to study gene expression. So we'll talk today about quality control, the role of pre-processing, different mapping strategies for alignment to the genome or the transcriptome. A new aspect of gene expression is the development of transcriptome, or the expressed genes, quantification and visualization of gene expression data. So the context of this session is there's a lot more detailed information on all of these aspects of RNA-seq. It's called RNA-seq, or transcriptomic data analysis. And we have 4 different courses. Transcriptomics 1, that talks about the basic processing steps. Transcriptomics 2, that talks about statistical analysis of the gene expression table. Transcriptomics 3, that talks about machine learning in application to data mining and classification of gene expression data. And Transcriptomics 4, that talks about single-cell RNA-seq. So when we talk about this kind of data, we're talking about the central dogma of molecular biology, which states that DNA is transcribed to RNA, which is then translated to protein. So what you can see here on this cryomicroscopy image is the DNA strand right here. It's kind of faded, you can barely see it. And then RNA polymerases that attach to the DNA and produce RNA. And then some of that RNA develops into mRNA, and that mRNA eventually is produced into protein. So here's the actual RNA polymerase. It's a protein that actually unwinds the DNA, opening it up and making it into a complementary strand of RNA. And you can see that it actually happens throughout this whole DNA as it is unfolded. And you can have a lot of these different segments attaching to the DNA, and eventually the ribosome also attaching to the mRNA to produce the protein. So this is what it looks like, in a diagram format. You have the DNA that is transcribed. It is transcribed to tRNA, mRNA, and ribosomal RNA, and all of these different aspects are important for the process of translation, where tRNA, transport RNA, brings in the different amino acids, and rRNA is actually folded into a ribosome, and then the translation happens of the protein, which eventually is assembled into its final form. Okay, Angena is asking a question. There was a question in one of the quizzes: how do we measure length in base pairs? If you could please explain the answer. Okay, that's a perfect question to put on the forum. Let's address that in the forum, as we already started speaking here about RNA-seq, so not to confuse everyone. Please put that in the forum, and I'd be happy to answer that. Okay, so one of the important aspects is that we are dealing with RNA that represents not the exact translated sequence of the genes in DNA, but actually represents a combination, different types of combinations of exons. You can see here that sometimes exons are skipped, sometimes they are arranged in different combinations, and we have really transcripts, not genes, that we are measuring when we speak about RNA-seq, because we are measuring the expression and the way this expression happens. So sometimes you'll hear the words isoforms or transcripts, and that's when people speak about the product of that gene. The first initial product would be how the gene was transcribed, that would be transcript, and isoform, because a single gene could really take a number of different versions, and those are called isoforms. So how do we measure RNA abundance? There are a number of different ways to measure RNA abundance. We can use PCR, and PCR can be designed around a primer, specific primer, that helps us check for whether that primer is there and how much of it is there, right? So after that is typically done here, as you can see, in a gel, and you can see how much of a specific amplified RNA is present. You can either check that it is indeed there, or you can also quantify how much is there. Also, another method of determining the amount of a known segment is to measure them using microarray, which is really just a technology to measure the quantity of light. And we can, again, do it around design probes. So the idea of measuring mRNA abundance or detecting that a specific transcript is being transcribed has been around for a while. These technologies allow us to focus on one known or a number of different known genes, transcripts, exons, or fragments. What it doesn't allow us for is to discover novel RNA, right? And because of this process of exon rearrangement, exon skipping, and what's called alternative splicing, we are limited as to how many actual probes we can put on a panel, as you see right here. And next generation sequencing becomes the preferred technology because we can really capture all of the variation that exists in a certain point in time inside the cell. And that is what we call the transcriptome. Right, so the transcriptome is the collection of transcripts inside the sample that we are studying at a given moment. So the next generation sequencing data takes short reads, and without amplifying any particular region or specific transcript, gene, or exon, it just generates short reads from random locations throughout a specifically designed probe that amplifies, for example, poly-A tails that are attached to mRNA. So that's how we can know that we are only sequencing mRNA from the host cell. Sometimes people also generate total RNA, and total RNA will include any kind of RNA molecules, and that those molecules can be poly-A RNA, tRNA, ribosomal RNA, mitochondrial RNA, so many different types of RNAs. There will be a lot of noise, but you will also be able to detect other types of RNAs. For example, RNAs produced by viruses inside the cells, or RNAs from transport RNAs, microRNAs, long non-coding RNAs, etc. But when we talk about transcriptomic data analysis, typically those are going to be poly-A. These are tails of amino acids that are attached to mRNA. One of the biggest challenges with this kind of sequencing is the need for processing. And so let's talk a little bit about how specific steps in preparation for mapping and after mapping can help us understand what is really going on in the transcriptome. So as you remember, there are a number of different types of data formats that we have to remember as we speak about RNA-seq. The FASTQ file that contains individual reads, those reads can be of various lengths and different qualities, and that is included in the FASTQ format. The FASTA format is typically used for the reference genome. So for example, we might have whole sequences of nucleotides, and those represent chromosomes or specific portions of those chromosomes if we're dealing with non-model organisms, and sometimes could also be used to store information on sequences of proteins, not in nucleotides, but in amino acids. Now, when we deal with these big chromosomes, for example, as we saw previously, the human genome has over 3.2 billion base pairs. Those base pairs are organized into genes. Genes consist of exons and introns. Those exons and introns are combined together in different combinations to produce transcripts. So all of that information is stored in what's called a GTF file, or a genome transfer format file. And that genome transfer format file includes the beginning and the end of each annotated or known genomic region. So when we talk about next generation sequencing, the next generation sequencing process in the lab takes genomic or RNA, genomic DNA or RNA, so the raw material, biological material. That material is shattered and shattered into short fragments. Those fragments have a specific step of library ligation when specific sequences are attached to the beginning and the end of each read. Those are placed into the flow cell. The flow cell is inserted inside the sequencer. PCR amplification is performed to amplify the material inside the flow cell. And then the sequencer detects which nucleotides are at which position inside each read. And that is the process of creating digital data. So once that digital data about individual sequences is created, now this is going to be cDNA, so whether it's RNA or DNA, these are going to be nucleotides from what looks like a DNA sequence. But in terms of now taking this data and applying it to study mRNA, what we typically do is, oh, let me go back here. Sorry. So what we typically do is we take a gene. That gene has specific isoforms, and we want to identify from the FASTQ file, the file that contains all of the reads that we've generated, we want to identify where do they map so that we can identify how different isoforms are actually expressed in the sample that we are studying. So our goal is to try to map the short reads onto individual exons and combine them together to understand what isoforms are being expressed. Okay, so the process typically would look like something like this. You have a sequence of DNA. There are specific genes on that DNA. Some genes are transcribed. And after they are transcribed, this is what we take. This is our biological sample. We then amplify the reads. I'm getting what happened here. We amplify the reads. All right, let me try and do this again. Maybe I was holding down some button. So the reads are shattered, adapters are ligated, PCR amplification to amplify the number of reads, and then the reading. Okay, not sure what happened here with the presentation, but this is the process of RNA-seq, and the ability to study the RNA-seq. Let me just go back here to this screen. Okay, so here we need to apply specific types of steps to measure this information that represents the level of transcription for transcript A and transcript C, right? So you can notice that gene B, for example, is not expressed. So it will not be present in the transcriptome. Gene A and gene C might be transcribed at different levels. And that difference in levels between different samples or different groups of samples is really what we are after when we speak about RNA-seq. Okay, so are there any questions about how the data is actually prepared before we do the analysis? Okay, So let's take a look at how this is done on the platform together. So please go to server.t-bio.info. Put this in chat. Okay, let me know when you're there. So I'll wait until at least a few of you are there. Okay, so a couple of you are there. All right, excellent. So a few of you are there. Please let me know as the rest of you arrive and are logged in. All right, excellent. So here we have the transcriptomics section. If you hover over this eye, you can see the description of RNA sequencing that we just talked about. And here you have a drop-down. Now, in the transcriptomics course collection, we actually use a couple of these examples, the PDX project and the cell line project. We'll start with the PDX project today because that is the basis of Transcriptomics 1. As you open this, let's take a look at the different sections included. You have the pre-processing steps. Those steps are important to process the individual short reads before they are mapped onto the reference. You have a section on error correction. Typically, this is used for some of the older sequencing technologies where it's important to address the individual point changes in the read based on the quality of that read sequence. Then you have this combined section that actually deals with mapping on genome, exon detection, and mapping on junctions. How do we know that two exons are spliced together? We have what's called a junction. A junction is a read that has the beginning of one exon and the end of the previous exon. So that's the whole section. It could be done either in individual steps or by algorithms that combine these three steps together. Isoform construction. So after we detect exons, we can actually construct isoforms by looking at similar level of expression across multiple exons that represent a single isoform. Once we detect those isoforms, maybe there are new ones, and those new ones are not represented in the genome transfer format that we use for the reference genome. So we have to add this new information to the GTF, to the genome transfer format that we use. And after we have that, we can map the reads onto transcripts and quantify levels of expression. Now, there are a variety of ways that you can actually go through these steps. If you look in the beginning on the start, you'll see how some of these different methods are linked under specific types of steps, right? So the logic of analysis is that we have to pre-process, then detect transcripts, whether we detect new isoforms or we just rely on the present GTF. We can then construct isoforms, or we can measure expression levels. Okay, the other thing actually that we can look at are the input files. So let me do this again. When you click on start, you can also take a look at what are some of the input files that are going to be used. And these are going to be SVL files. So as you go through Transcriptomics 1, you'll see what's inside those files. So as we will do first the pre-processing, we talked about adapter ligation, and those adapters typically are recorded in a database. Trimmomatic is a job that actually looks through all of the possible adapter sequences and removes them to clean up the reads. This is a computationally intensive process because there are many different adapter sequences. And so to go through all of them, it takes a considerable amount of time, especially if you have large, FASTQ files. After that, you can reduce the number of unequally amplified reads. So PCR amplification happens right here. That bridge amplification is designed to make sure that you have enough material, but PCR can be typically biased because of the different sequences of reads. So some reads will be more amplified and some will be less amplified. to adjust for that difference in amplification and remove reads that are abnormally replicated many, many times, there's the step of PCR clean. As I mentioned, you can then choose basically between the steps of individual mapping on genome, exon detection, mapping on junction, combine them together in HiSat or top hat, or directly skip over to mapping on transcripts, right? So let's forget about finding new isoforms, new combinations of exons, directly go to mapping on the transcriptome. So bow tie to T, just like bow tie to G stands for mapping on G genome, bow tie to T stands for mapping on transcripts or the transcriptome. Same process. So if we connect these two, now we can quantify levels of expression. How do we quantify levels of expression? It seems like a simple task. Just count the number of reads aligning to an exon or aligning to a region that we call a gene. But it turns out that they don't align in an equal way. So now we have to think, well, how do we actually address this problem and give it a number? At the end, we're interested to know how many reads map onto a specific gene, right? So this is where the process of RNA-seq using expectation maximization comes in, a complex algorithm that is designed to produce a table of gene or isoform expression. Okay? Then we have some downstream analysis steps. Typically, this includes comparison of samples that are representing different conditions, and that would be differentially expressed genes, DESeq2, as an example. Let's skip that for now and click on End to see what would be the result, okay? So let me know if some of you have been able to go through this demo pipeline. Just type in yes, so I'll know that at least a few of you have gotten to the same result. And if not, if you have any kind of questions, then also let me know. Okay, has anyone been able to make this pipeline? Very simple pipeline, pre-processing steps, mapping on transcripts, quantification, and then end. Okay, so some of you have done it. Excellent. Okay, so after we get the result, right, you clicked on end, you can see that we have a number of different outputs. This is important to remember that each pipeline will include a number of different outputs. It's not just a single output that gives you all the answers, and so you have to look at what else is included. So we'll talk about some of these outputs in the future. Let me ask you a few questions here to make sure that everybody understood what is going on. Okay. So which of the following are pre-processing algorithms? 1, top hat and DESeq. 2, PCR clean and trim-o-matic. 3, bowtie 2 or BWA. 4, edge R or DESeq 2. So which one do you think is a pre-processing or R? Okay, PCR clean and trim-o-matic. Excellent. Okay, so all of you got this correct, right? So we have pre-processing, trim-o-matic, and PCR clean. Trim-o-matic cleans technical sequences. PCR clean removes PCR duplicates. Okay, so let's do an overview. What would be the step number 1? That would be pre-processing, right? Then after we do pre-processing, we do mapping. We map those reads that have been cleaned up and prepared onto either the reference genome or the reference transcriptome. And what would be after that? The step is quantification of expression levels using estimation maximization, for example. And it's important to remember that that step can include a number of different outputs, including different ways to measure gene expression. We can use different types of normalization techniques like FPKM and TPM. We'll talk about those in detail in Transcriptomics 1. Or read count. Read count is the basic or raw output where we just give you a number of all the reads that have aligned to that transcript or to that genome. Here's an alternative example, a more expanded pipeline. This expanded pipeline, instead of going directly to bow tie to T, will include the step called HiSat2. HiSat2 will map on genome, detect exons, and then map reads on junctions, right? So we're gonna look for the evidence that two exons are spliced together. Once we know that they have been spliced together, those could be constructed into isoforms. So we have a number of different exons, and that number varies between different isoforms. They're aligned together in specific ways, so we know how their order is or what their order is, and we now can detect new isoforms. Those new isoforms need to be added back into the genome transfer format that we use for annotation. And then we can map reads onto those transcripts that we have identified to be present in a given sample and prepare an RSEM expression table. Okay, so pre-processing, we have removed or cleaned up the different reads that we have. And it's important that we also remove bad technical artifacts and bad read quality, right? And the outcome of this is that we will have a higher likelihood that the reads will align to the right location on the genome, and we will have a higher quality, higher confidence in the final calculation of levels of expression. For mapping on the reference genome, we have a few different strategies that we discussed. We can map on the set of known transcripts, map on genome and potential identification of novel transcripts, or a combined strategy, right? So one direction is when we have this combined strategy with HiSat, or we have individual steps that we can take to map on genome and then map on junctions, or we can just map on the transcriptome. Right, so here we have identification for the best place for a read alignment, which is important. We have to identify junctions, and we also have the option to visualize those map reads. And of course, all of these steps are fairly complicated. Okay, finally, we want to quantify expression levels. The quantification of expression levels is important because it actually will give us the data that we will work with downstream in our next downstream steps of analysis. So when we do quantification, there are a number of different ways we can quantify that data. Sometimes it is normalized in different ways, and that different types of normalization is something we'll discuss in Transcriptomics too. For quantification, we have a number of different tools. For example, RSEM that we talked about, it quantifies gene and isoform abundance from single-end or paired-end RNA-seq data. There's Sailfish, a novel method that is called alignment-free. It doesn't actually use alignment. It maps onto a database instead of using the genome sequence and the GTF file. And also there's another method called HT-seq. So a number of these different methods actually do the same thing. They basically quantify gene expression. So let's take a look at what is the output of these different steps. The output of these different steps is what's called a table of expression. And inside that table of expression, you have individual genes, right? Genes will, if it's a human gene, will have the beginning ENSG and then some kind of an ID at the end. And we can quantify its level of expression by, for example, looking at a bar plot. This is called a bar plot. So here you can see inside of our, let's take a look at this output from the pipeline. Okay, so here you have all results, expression genes, FPKM not filtered, meaning that even insignificant samples are not going to be filtered out. FPKM, so these are the filtered, right? So let's download this one just to take a look at what's inside. And we can open this in Excel. Now this data set that we're dealing with, it is explained in detail in Transcriptomics 1. It is a study of breast cancer in animal patient-derived xenograft models. What that means is that a tumor has been implanted into an animal, a mouse, and as a result, the mouse contains both the mouse genome and the human genome. Okay, so here you can see ENSG, which stands for ensemble ID of a human gene. Okay, and this is some kind of an ID. And it's expressed in triple negative breast cancer samples. So there are 2 samples that are triple negative, and you can see that they have a level of expression. Okay, so what is its level of expression? This is in FPKM units. So these are called fragment per kilobase per million, which accounts for how long the gene is. This specific gene might be of different length from another gene, let's say this next gene. And so statistically speaking, reads are more likely to land in a longer gene than a shorter gene. And so that's why this accounts for the length of the gene. It also accounts for the total number of reads, because the total number of reads between the different samples could be completely different. And so if we want to use them to study gene expression abundance, how many genes are expressed in a particular sample, in the context of this study, we can account for the total number of reads that is included in each sample. Okay, so to look at this gene, we can say that in triple negative samples, you can see TN for triple negative and ER for ER positive. So for triple negative, a couple of samples have an expression over 20. Let's take a look at another example. Okay, in this example, you have ER. So 1 sample from ER, 1 sample from triple negative. But how high is the expression, right? The scale here changed. So instead of 20, this is 1.2, and this is below 0.6. So would you say this is a significant gene when we talk about this study? So yes, if you think it's significant. No, if you think it's insignificant. Okay, Juan says it depends. What does it depend on? Anyone else? What do you think? Is this gene significant to count for or not? Okay, as you're thinking about it, okay, so he's saying depends on the data set, maybe 0.6 difference is significant. Okay, so that's a good point. So let's take a look. How many genes do we have in total here? So if we scroll all the way down, we have over 3,300 genes. And out of those 3,300 genes, we find one gene that looks like this. It is below 1.2, right? And really, in my opinion, it is going to be insignificant, because even if we think that this represents a difference, the difference is only between 2 individual samples. It's not between groups, right? Because we don't have an average in this group. We don't have an average in this group because all the other ones are 0. So typically, this would be called an insignificant gene, because we can also compare it. Let's take a look at another example, and just to see the scale, okay? Look at the scale. There's 25,000 is the high level. The low level right here is 1,392, right? So the scale plays a role. The number of samples that have expression plays a role. And the normalization technique plays a role as well. But it's not as simple as just simply looking at those numbers. This is just to illustrate what types of examples do we have. Now, ENSG stands for human, as I mentioned. Let's scroll down. What about ENSMUS? What do you think that stands for? What kind of a gene? Okay, so Juan here is saying mouse gene. That is correct. So we have, as I mentioned, a human tumor implanted into the mouse animal. So we have two different genomes. What does this kind of a combination give us? It actually gives us this unique ability to separate what goes on inside the tumor and what goes on in the microenvironment of that tumor. The microenvironment consists of primarily human sample, or sorry, mouse. The microenvironment is the mouse because that's the animal where it's implanted. The tumor comes from the human. So we can separate and see how the tumor interacts with the microenvironment. Okay, so these are some of the examples. If we are looking at transcripts and not genes, we will have what's called ENST. Sometimes you will also find a transcript that is called XLOC, and that is a novel transcript. It has not been annotated, and typically those will be found when you do isoform construction. In your Transcriptomics 1 assignment, make sure that you look through all of the genes. Make sure that you look through all of the files. Some of them might be insignificant, therefore filtered out, so they're not going to be in the filtered tables. Okay, any questions about what we talked through today? Okay, so if there are no questions, if you do have questions, please add them to the forum. I'd be happy to focus on those next time. Let's briefly think about what do we do after we get the table of expression. So the table of expression represents samples, which is going to be column names. So this is the name of a sample. A lot of times, okay, a quick question from Manal. How do I go back? Just right click on the button that you selected. If you right click on it on your mouse, you will go back. Okay, so the sample name, a lot of times the sample includes the previous steps. For example, here you can see no adapters, right? That signifies that this was done with Trim-O-Matic. No PCR, so that means it was also processed with PCR clean. And you can also see that these are pair end reads. And then here you will have gene ID. So that gene has what we call an expression profile, which is the collection of expression values across the samples that you have. Okay, in this cell, each individual number in the cell represents the level of expression. And that level of expression could be in a number of different formats: FPKM, RPKM, TPM, or read counts. And it's important to remember what it is because of downstream analysis. Now, what do we do with this table? As you saw, we have 3,000 genes. Sometimes we have 20,000 genes. One of the ways that we can summarize this data is to look at a box plot. So a box plot typically represents the bulk of your data, or the interquartile range, that shows you some of the major statistical properties of the data. If we just do a bar plot for the gene expression table that we just saw, it will look something like this. Why is that? That's because this type of transcriptomic data in a normal kind of direct output that we will look at is going to have very large numbers. So these are very large numbers. You can see close to, tens or even sometimes hundreds of thousands. And there are going to be very many small numbers that are going to be, up until 10 and 50. And so the bulk of your data is actually down towards the bottom of the scale. And it's very difficult to see what is actually going on. How do you compare these samples to each other? And so one of the ways that you can think of looking at the majority of the values instead of the outliers that are more prominent is by transforming this to what's called log scale transformation by transforming it into logarithmic scale. When we transform this kind of data into logarithmic scale, it helps us see better the bulk of the data without changing the actual values, right? So we always have a path to go back to the original data because this is a simple log normal transformation. Okay, so then one of the ways that we can study this data is by also looking at the histogram. And the histogram can be useful for us to do what's called quantile normalization. The quantile normalization is the ability to make those samples comparable. Why do we need to make them comparable? As we look at the gene expression table, we want to understand how these individual samples represent a condition. Okay, so here's another example. We have here two different types of breast cancer samples, not triple negative and ER positive anymore, something else. But we want to understand what are the differences, what are the differentially expressed genes between one condition and another condition, the green ones versus the yellow ones. To be able to do that, we need to assume that the samples within each group are actually representative of that condition. That means if a gene is expressed at a level of 10 in this sample and at a level of 1000 in this sample, that means that maybe these two are not really representative of that condition. Maybe they're so different that I cannot use them to compare those two different conditions. To eliminate some of that variability, quantile normalization actually takes the noise, which are different outliers and genes that have unstable levels of expression throughout the different conditions, and normalizes the noise. And as a result, you get a higher signal sensitivity that represents that condition, okay? So quantile normalization is a technique that you will see in Transcriptomics 2. The result of quantile normalization is actually making the data, so here you can see before quantile normalization, here you can see after quantile normalization. Here you have samples from 50,000 all the way to 10,000, or let's say 15,000 in one condition, right? So this sample is going to make it difficult to understand the level of expression for this condition. But here you can see the data much more comparable, and now you can actually study the difference between the conditions. In this case, there's actually no difference between these two conditions, no statistically significant difference, even though here you might have thought that there might be because of this sample and this sample right here. This is important for especially the highly expressed genes, okay? And I suggest for all of you to try and go through Transcriptomics 1 and 2 to understand both the process of preparation of the data, or what's called processing of RNA-seq data, and exploratory analysis, when you're actually able to look at the gene expression table and understand how to prepare it for downstream analysis. So let's do a quick overview. Pre-processing steps that we talked about, cleaning up the reads, cleaning up the raw material, essentially is pre-processing. The next step, mapping. Take those cleaned up reads and align them to the reference genome to identify transcripts and express genes. Quantification. And after that quantification, it's important to measure how the expressed genes compare between conditions. So typically there's an additional step called differential gene expression. Differential gene expression will help us understand how genes are differentially expressed between groups of samples. Okay, so again, we have the transcriptomics section right here. And for downstream analysis, a number of different tools down there. One of them is called differential expression. You can also see what differential expression is. So differential expression helps us study variation between conditions. And when we do that, we can add that DSeq step at the end of a pipeline. So this area right here for differential expression includes a number of different algorithms that essentially help us identify only those genes out of all of the genes that are differentially expressed between two conditions. Okay, so what did we learn today? We talked about the biological role of transcription, methods of quantification for mRNA, how to use next-generation sequencing to study mRNA expression, and how it's important to understand the steps of analysis of next-generation sequencing data when we study mRNA expression. We talked about pre-processing, mapping, and quantification, the meaning of gene expression for sample variability, and the biological interpretation. And this is important because from DNA, we went to RNA. And that RNA is what we study in RNA-seq or transcriptomics. In our next session, we'll talk about some more advanced methods for analysis of this type of data. This is a very detail-rich data, typically contains a lot of patterns that are not as simple as differentially expressed genes, even though differentially expressed genes are the easiest to interpret. So in our next session, we'll talk about data exploration using dimensionality reduction and clustering, classification and discriminant analysis for labeled data sets, as well as unsupervised and supervised machine learning. So again, thank you for joining today. If you have any questions, I'd be happy to answer those. If not now, and you come across those questions, for example, when you do Transcriptomics 1, 2, 3, and 4, you can post that on the forum. We'll be happy to address those in our next session. To give you a sense of where we are in the program, we're approaching the middle of the program. So we've covered today, we finished the third one. Next time we'll talk about machine learning, and we'll start going into examples of different projects soon. Okay, Juan is asking the server, could we use to practice different pipelines? Yes, that's exactly the purpose of the server. I do recommend using it to do it in the context of a course, okay? June Meng is saying, could you send the invitation calendar one day before the event? Okay, so here you have all of the dates. It's always at the same time. So please just add it to your calendar to always have it in advance, so you don't have to remember in the middle of the day that we have a session. The session email is just really a reminder for you to have on your calendar in front of you close to that time. You can also go to the syllabus link that Beepsha just added to the chat. And on that syllabus, let's take a look. We actually have all of these dates posted. So if you go right here to menu, syllabus, under syllabus, you'll be able to see all of the upcoming sessions. Okay, so what we did today, and actually also shows you the material that we asked you to cover before the next session. So I know that some of you are probably feeling a little bit behind. You will have time to complete all of these courses, so at least try to go through some of the introductory lessons in these that we post right here that will help you understand better and have the ability to practice some of these pipelines, understand the outputs, understand the context of the data sets that we use, right, is really helpful in between the sessions. So our next session is going to be machine learning for gene expression data. If you can finish Transcriptomics 1 and 2 before the next session, we went through a lot of it today in the session, but if you have more time, please complete that. And then we have additional courses that will be available after this session next time. Okay, does anyone else have any other questions? Okay, Shipra is asking how to interpret output results, output files. Perfect. So as you saw, we have a gene expression table. That gene expression table has specific genes. Each gene if you search for that gene, has a specific function. Sometimes that function has been studied and is known. Sometimes it has not been studied and is not known. So here, for example, this first gene that we looked at, okay, their server is down. So let's look. Okay, so here's another portal for this gene. So you can look up the role of this gene by a common name and what it does. This gene encodes a member of the cytochrome P450 superfamily of enzymes, etc. So the problem is that you have thousands of genes. And once you find a small subset of genes that are significant, for example, that are differentially expressed between the two conditions that we have here, then you can start making assumptions about what the total functionality of these genes might be. A lot of this material is actually covered in Transcriptomics 2, and we'll touch on this in our next session. We have different methods of annotation, how to take collections of genes and assign them with common names, just like this is a common name, and function, which is this, as well as associate them with specific pathways, because genes work in pathways, and what's called gene ontology. So gene ontology terms, as you see right here. By grouping genes together into groups that have similar functionality or that have similar pathways, they mean that the pathway is activated, we can start thinking of their biological significance for a specific condition. A very useful resource, again, described in detail in Transcriptomics 1 and 2, is a portal called David. Okay, this portal allows you to take lists of genes and look at how these genes are known to behave in the cell in different locations in the cell, different types of functionality, and different pathways. Okay, so something is happening today with David, looks like maybe. But I'm sure it's not going to last too long, so you can take a look at this link right there. Okay, are there any other questions? Okay, there's David. Database for annotation, visualization, and integrated discovery. So we'll talk a little bit more about this next time as well. Okay, there are no more questions. I am looking forward to seeing you again next time. And remember that we have the free webinar. So please come to the webinar to learn about additional data science and bioinformatics programs that you might be interested in, to learn about the different proteins, structure and function of different proteins, as well as some data science skills that include some coding in R and Python, very relevant for our next session when we'll talk about different machine learning and data mining techniques for RNA-seq data. Thank you for joining today. We'll have the recording posted soon, and I'll be looking forward to seeing you again next time.